Thanks, Finn, and welcome to everyone to this webinar. And as Finn alluded to, the Freshwater Quality Programme has been joined by a new project called CLEAR-Neagh, and this is the project we're going to hear about today. So that brings the total projects within this programme up to 6, and we'll be hearing from the other projects in the next few months. So we've CLEAR-Neagh joins Quantum and Pacific. Quantum will be hearing from next month. And Finn, if you want to just forward the slides. Yeah, and LTSE-FE is in May. MOT for Rivers we heard from last month, and EcoMix will be hearing from again this coming few months. So as this is our, so I should say, I was going to say about the programme, but those of you that joined before, basically the ultimate aim of the programme is to use science evidence to inform improvements in UK freshwater quality. And there's a number of ways that you can get in touch with us. There's our email address, and there's our website, and you can follow us on social media. So as Finn said, all our webinars are recorded and they are on our YouTube channel, which you can access through our webpage. So if you want to catch up with any of the others. And as I was saying, this is our 3rd year of webinars. So we're hearing from the projects that started 3 years ago for the 3rd time. So Quantum will be in April, LTSE-FE in May, EcoMix in June, and Pacific in July. As I said, MOT for Rivers was last month, and we'll have a break for the summer, and then we'll have some other speakers in our autumn session as we normally do. So without any further ado, I'll pass over to Susheel, who is leading the CLEAR-Neagh project, and he'll outline the project for you. So Susheel.
Thank you, Pippa. I'm assuming you can see my slides. Please let me know if you can't.
Yeah, it's all okay.
Brilliant. Thanks a lot. So, morning, everyone. Apologies at the outset. I speak His Majesty's English in my own Indian way, so things don't get lost in translation. I'm here to talk about CLEAR-Neagh, which is our project on Lough Neagh, specifically Catchment-Level Environmental AMR and eDNA Reconnaissance. And essentially, my talk is set up into an introduction to the ecological issue that is around Lough Neagh, a little history in time, and the invasive ecosystem engineers, an overview of the project and what we do, especially with eDNA or environmental DNA, both in our group and also in what we plan to do in this project. So first and foremost, though, I must thank our funders, so DERA and UKRI NERC, also the program led by Water at Leeds and several people, my team, and our collaborators, the Lough Neagh Partnership and Biota Trace, who are key for this particular project being undertaken. So Lough Neagh, why do we actually care about it? Well, for those of you who are not familiar with it, it is the largest freshwater lake in the UK. And in fact, it's an invaluable resource providing over 40% of Northern Ireland's drinking water. Now, Lough Neagh as a catchment was designated as a sensitive area all the way in 1994, and there are over 23 protected features. The loch itself, apart from drinking water, is useful for recreational activities where you have open water swimming, but also for several commercial activities, significantly for eel and pollen fisheries. And in fact, Lough Neagh is one of the largest eel suppliers for Europe. Now, the catchment of the loch is approximately 43% of the land of Northern Ireland, and it's a freshwater body that is very much of international importance, and it has several main rivers feeding into it. Now, in 2023, this became an unprecedented issue where it was highlighted that there were several large-scale algal blooms that were found not only immediately within, but also were observable enough from satellite imagery. And you can see here on the left various samples that were collected across the lake in these falcon tubes, which show a range of what the algal bloom capacity was like. And this study done by Reed et al. From Queen's University Belfast, in fact, highlight in the satellite imagery in the bottom left the scale of these algal blooms. And this picture captured by Peter Harper from the Lough Neagh Partnership shows you what the real life scenario was like. So this was something that was unexpected and completely unprecedented. Now, what are the potential causes for something like this? And off the bat, there are three primary causes, among others, and these are typically eutrophication, climate extremes, and invasive species. Now, the concept of eutrophication, which is the influx of excess nutrients such as phosphorus or nitrogen, is typically a phenomenon that is seen in many catchments that have high agricultural inputs. And in fact, this is a story that is as old as time. So even back in 1973, Wood and Gibson reported that eutrophication could potentially be a problem in and around Lough Neagh, where they started observing changes in phytoplankton or microbial concentrations in the lough itself, especially in alignment with the farming timelines. Now, this was also subsequently reported in 2007, where the degradation of the water quality in Lough Neagh, especially from diffuse nitrogen fluxes in phosphorus-rich catchments, was highlighted by Linda Bunting and colleagues. So outside of eutrophication, there is also the aspect of climate change. And whether you believe in climate change or you don't, data suggests that, in fact, this is a real phenomenon. So the surface temperatures, and this is data that I pulled for Northern Ireland, and the surface temperatures since 1995 have increased by at least 1 degree Celsius. And this you can see in the graph on the top, where from 2007 all the way to now 2025, it has an upward trend. Similarly, we've had some of the highest recorded rainfalls in recent years, and 2023, especially when the algal blooms were at their highest, also had the highest recorded rainfall in the history of Northern Ireland itself. Now, a third aspect, as I mentioned earlier, is the potential invasive species. Now, invasive species are those species that are typically not native, but those have been brought along by accidental factors or other mechanisms. And in fact, one of the most well-known invasive species in Lough Neagh is the zebra mussel. And these mussels were first reported in 2005 as having been found in the lough. And since then, they've essentially taken over the endogenous population, which is the swan mussel that is typically found in Lough Neagh itself. Now, these zebra mussels are what we call filter feeders. They essentially sit between the interface from the water and the sediment, and they feed on the phytoplankton and any other potential organic matter debris, and it increases the clarity of the water. Now, what happens when this clarity of the water changes, as in it becomes clearer, there is increased light penetration. And the light is a very important factor for cyanobacterial reproduction and blooms. And when you have more light, you have additional eutrophic situations. You have these algal blooms happening because of a contribution of all of these three events. Now, we visited the lough a couple of weeks ago and, in fact, took in its very serene beauty. The waters were fairly high, as you can see here on the left, tree virtually submerged halfway through. And part of our team here on the right, Linda May Hawley and Lauren, along with Peter Harper from Lough Neagh Partnership, we walked around the catchment to understand the scale of the issue. Now, while we did this, we did come across the many mysteries that the Lough potentially has, and I'm still yet to unravel some of these mysteries, but I look forward to doing that. What you are seeing on the right, though, is an inset of an image of the swan mussel, which is the larger shell, and then on the inside is a zebra mussel. Now, this is not a typical natural occurrence. I put the zebra mussel in there to compare the size and show you the scale of the problem that we might be potentially facing, because although tiny, they are numerous. So the shells that you see in the image on the right are essentially a lot of shells from the zebra mussels around the lake. And when we in fact walked around the lake, we found a lot of these shells and were immersed in them. Our shoes were. And the extent of this picture, all the way to the top of the picture, these are all shells around the lake itself. So the problem is quite huge. We found other invasive species. You might recognize some of these in this site, but I believe these do not pose a serious threat. So the triad or the trifecta of eutrophication, invasive species, and climate extremes are likely some of the causes that are leading to these algal blooms. And in fact, this was captured quite well by Reed et al. Again, where they posited that other sources, including cattle and poultry and septic tanks, are key causes as well that might be leading to algal blooms in the future. What is important to highlight is there is also a lot of the legacy phosphorus down in the bottom of the sediment. So in this image, you can see the P legacy phosphorus that has been stored in the sediment that is released over time. And Reed et al. Reported basically that they found a lot of toxins in these algal mats that are potentially detrimental to human health. Now, the respective authorities released a report in 2024, and this was led by DERA, where they highlighted that in the Northern Ireland water quality metrics, phosphorus pressures were quite high, and typically these came from either agricultural sources or wastewater sources, septic tanks. But in fact, the Lough Neagh pressures were specifically around increased temperatures and the zebra mussels, and also the legacy phosphorus release. They also put forward a 37-point action plan, of which 15 are delivered. I'm not going to go through these. You're welcome to read the report. But what is important is some of these progressing action plans that still remain are associated with the focus on long-term sustainability and eventual restoration of the loch itself. So this is where we come in. So CLEAR-Neagh is designed to basically trace the origins of microbial populations across the loch and to support evidence-based restoration. Now, we know the loch faces a nutrient sediment pollution issue. However, current monitoring efforts cannot necessarily differentiate what are potentially human sources or what are agricultural sources. And importantly, we believe there is a need to capture both temporal and spatial dynamics of pollution, and this was set out in the report as well. So our solution to this problem is to integrate key genomic methodologies such as microbial source tracking and eDNA alongside existing hydrological models. Importantly, we would also be bringing in autonomous and high-frequency sampling across the catchment to produce risk maps and toolkits for restoration and stewardship. So what we're trying to do is bridge the gap between pollution pressures and evidence-based management. Now, before I go into the specifics of the project itself, I just wanted to introduce the concept of environmental DNA. For those of you who are not familiar, this is the genetic material that is released into the environment, whether it's coming from water, soil, air, trees, animals, humans, and the sources could include anything like cells or feces, mucus, carcasses of microbes. What we can do is we can use the genetic markers in these in this environmental DNA to identify where the pollutant sources are potentially coming from, whether they are coming from agricultural runoff or water discharge. And it also allows us to capture a snapshot of the biodiversity without directly sampling the organism. And this, in fact, is a representation of the potential sources that might be coming into Lough Neagh itself. So you could have your urban human sources, such as wastewater treatment plants. You can have your wildlife sources, be it birds or other animals. You can have recreational sources, humans when we swim in the lake, or you have boats coming in, and you have the agricultural sources, among others. So we would use two different methods, as I mentioned earlier, and one of them is the microbial source tracking. The genetic markers of the microbes that are found in different animals, for example, whether it's cows or humans or birds, allows us to differentiate where the pollutant sources might be coming from. So there are very key specific regions that we can identify and say, well, this microbe came from a pig or this microbe came from a dog. And using this, we would be able to say where the pollution is potentially coming from. Additionally, we would use antimicrobial resistance profiling, and this is to look at any gene that is found in a microbe that might confer resistance against antibiotics that we use for our human health purposes and treatments. And the proxy for looking at this is the INTI1 gene, so the integron gene, that is a very highly reliable and well-cited proxy for anthropogenic pollution. What it allows us to do is it provides an early warning radar for emerging environmental pathogens, and it helps us identify potential risk of AMR genes being transferred to ecosystems. There are other ways of doing this, and both at UKCEH and our group, we are involved in national scale monitoring programs where we look at freshwaters and soils, including even honey, to understand not only the diversity of the microbes and other higher order animals, but also their biogeography, their functions, and environmental drivers. And we do this using what we call a one sample, many analysis methodology. Well, what this entails is collecting a sample from whether it's freshwater or soils. In this case, we would be collecting the sample from the lakes around the shoreline, and then extracting DNA from these. And these would subsequently undergo multiple analyses. The first of which would be a quantitative PCR screen, so polymerase chain reaction screen, looking for fecal markers where we can then identify what percentage of that DNA is coming from which likely source, whether it's a poultry source or whether it's a cattle or whether it's human sources. Additionally, we can use the same DNA to test using a metabarcoding approach where we're looking at highly conserved genes in both prokaryotes and eukaryotes, and we can identify what taxa are present there and what does the overall community look like. Importantly, and one of our USPs here is using shotgun metagenomics. Because we are collecting and extracting any and all DNA from these samples, we can understand what are the functions that are being encoded by the various microbes in the system, and importantly, all the different AMR genes. So we're not just limiting ourselves here to the INTI1 gene that I mentioned earlier, but in fact profiling the entire AMR gene catalog in the system. So this brings me to how CLEAR-Neagh is set up. So along with our collaborators, both the Lough Neagh Partnership and Biota Trace, CLEAR-Neagh is set up in 4 work packages, which I will go through in detail in a bit. But work package 1 is taking existing data and models and integrating these to map catchment inputs, which will then be used to identify locations or high-risk locations where we can deploy our spatiotemporal sampling to look at catchment pollution sources. Work package 3 subsequently will characterize the microbial communities, the AMR in the systems, the antimicrobial resistance, and also the biodiversity. And the most important of them all is the work package 4, where we will actually translate all of this knowledge to help with data stewardship and stakeholder engagement. So work package 1, as I mentioned, is essentially using different models around human population densities, whether it's wastewater treatment works and CSOs that are available in a catchment, looking at livestock density models that we currently already have, and land use information, and also underlying hydrology, information tracing the subcatchments to the lake. All of these would be operationalized to create layers and proxy maps where we can focus on where the most pollution is coming from and target our sampling around those areas. Work package 2 subsequently uses this information and then deploys a targeted multimodal sampling network. I realize this is a busy slide, but what I'd like you to focus is on the right. We would be deploying shoreline sampling, so the yellow dots around the shore, and also sampling up in the tributary, so the rivers as well, to identify where pollution sources might be coming from within the catchment itself. We will also have kiosks, so these the green units that you're looking at, and I'll show you an example of that in a bit, which we will deploy to collect automated sampling, especially in a storm-triggered event. Now, sampling is a very key event, and despite my colleague here, Lauren, smiling about it, it can be challenging, especially when you're pushing through lots and liters of water through a small syringe and a filter. So we've teamed up with Biota Trace, who have an autosampler that is as simple as assembling a syringe and a Sterivex filter, here you see on the right, and then deploying it using their automated sampler. Now, these can be dipped straight into the water or dangled over a bridge. And the utility of these samplers is you can have reproducible methods where you can monitor the amount of volume that you're sampling and also control the amount of pressure that you're applying, so you're not necessarily damaging the filters. Subsequently, the samples would be preserved and then brought back to the lab. And one USP of our sampling method, the autosamplers, is this is mobile. So we can literally put it on the back of a van and drive around the lake to collect our samples. The other sampling methodology we're using is this unmanned surface vessel that can be pre-programmed ahead of time in terms of coordinates, and then it would go around and sample, whether that's closer to the shore or further into the lake, or whether we need it when there's a particular adverse weather event. And finally, the kiosk, which essentially is a box. So due to IP reasons, I can't show you what goes on inside, but you will have to take my word for it that these will have about 40 autosamplers in any one instance, which will be used for work package 2, where as I mentioned earlier, we will do a seasonal shoreline sampling, a longitudinal sampling, and also the USP sampling. Work package 3 then takes all of these samples, extracts all the DNA, and essentially identifies the taxonomic and functional quotient of all of the information from this to identify traces of pollution, construct networks, and get at AMR. So as I mentioned earlier, that work package 3 encompasses the quantitative microbial source tracking, the taxonomic community analyses, and the gene profiling. Last but certainly not the least, any data that we produce is useless without translation. So in work package 4, we would be converting all of this into accessible, interoperable tools for the regulators and water managers and the communities, and directly feed this into the Lough Neagh Science platform, which will deliver actionable evidence that can be used for downstream decision making. Work package 4 is also built on FAIR principles. It's findable, accessible, interoperable, and reproducible principles. So all of the workflows that are developed for these and any data will be openly available to anybody, any parties of interest. And all of these will use a harmonized metadata schema that can be seamless and pulled into the Lough Neagh Science platform almost immediately. Subsequently, the goal is to deliver a practitioner toolkit, whether it's guidance or minimum threshold values, newsletters, or decision pathways that can be used in the stewardship of the loch. So the CLEAR-Neagh integration engine is designed to be iterative. So the catchment modules can dictate where we could sample. The sampling will then eventually feed into the molecular analysis, and subsequently this feeds into the translation efforts. And this is very much a circular process that can be fed continuously, and you could enter at each point as needed. The long-term goals, of course, of CLEAR-Neagh, in collaboration with the respective regulatory bodies and our collaborators, is to deliver long-term stewardship for Lough Neagh and Northern Ireland. And our goal is to try and equip the communities and the regulators with definitive evidence to trace pollution, potentially mitigate risk, and also restore the ecological resilience. With that, I'd like to thank you all very much for your attention, and I'm happy to take any questions. Thank you.